Hello listeners, and welcome to the Dr. GPCR Podcast, where we bring you conversations with leading experts in GPCR biology. In each episode, we dive into the career trajectories, discoveries, and groundbreaking research shaping the field. The podcast was born at the height of the pandemic with three key goals: to share the latest discoveries, give researchers a platform to showcase their work, and inspire the next generation of scientists to explore this fascinating field. Since our launch, we've recorded over 160 episodes featuring incredible guests like Dr. Brian Roth, Dr. Robert Lefkowitz, Dr. Fiona Marshall, and many more. Our mission is to unite the global GPCR community, encouraging collaboration to improve human health through a deeper understanding of GPCR biology. Thanks for joining us in this exciting journey, and now let's dive into this episode.
All right, here we go. Hello, everyone. This is Yamina from Dr. GPCR, and we finally made this call happen for various reasons, link issues, calendar issues, but today I'm very excited to have back on the podcast Doctors Tom Sakmar and Ilana Kotliar. Now, you're a doctor. I'm very excited to have you on, as well as Tom. And today we're talking about new tools that were developed in Tom's lab at Rockefeller University to better understand GPCR-RAMP interactions and how those influence GPCR biology. So welcome back.
It's great to be back.
Thanks, Yamina.
Good to have you on. So where should we start? Who came up with this idea of looking at GPCRs and RAMPs in the lab?
Oh, well, that's an interesting story. It goes back to my interest in family B GPCRs, which goes back 30 years because I was sitting in my postdoc lab at MIT, and I got a call from Bruce Merrifield, believe it or not, on the phone, an actual phone call. And Bruce Merrifield, who I had known but had never spoken to before, said, "Tom, I hear you coming to Rockefeller to be an assistant professor. I know you work on GPCRs. I hope you're willing to work on glucagon receptor, which is my favorite receptor, but I also am interested in GLP-1 receptor and other family B receptors." And so that led to our lab working on glucagon and family Bs, and we've done that since the past 30 years. And beyond that, there was a particular grad student called Emily Lorenzen who wanted to work on family Bs and was aware of the RAMP story with CGRP and amylin receptor in those early days. She had just come after the amazing paper by Steve Ford showing the discovery of RAMPs, which is in around 2000 or so. And she said, "I'm not sure I want to work on one receptor at a time. Is there a way to broaden out my project?" And I said, "Well, this is potentially interesting because I think RAMPs might be more important than just involved in chaperoning one receptor. And there's this interesting story with the amylin receptor where they're affecting pharmacology. So let's broaden it out." And it so happened that at the same time, I had some connections, and we had a couple of fellows who were working on things, and so it all sort of expanded from there. And Emily Lorenzen made this first attempt to do RAMP receptor interactions in a multiplexed way with people in the group, and that led to connections that expanded to Karolinska. And without going into too much detail, this happened because at some point we decided to evaluate, meaning Emily and I evaluate the multiplexing methods that were available to study protein-protein interactions in bilayers. And there were several competing methods, but we came upon this Luminex suspension beta ray assay, and she said, "This looks interesting." And I said, that's kind of cool because at Karolinska, where I was doing a sabbatical at the time, there was a lab who was an expert at that. So she reached out to Jochen Schwenk at SciLife Lab in Karolinska, and we started a pilot project with various people in the lab, and it seemed to be promising. And that whole initial family B, the secretin receptor family pilot project worked. That was published. And as part of that paper, Ilana had just joined the lab as a rotation student, and she was involved with that paper and said, I really love this project. I want to take it forward. And then it sort of expanded from there. Because the beauty of multiplexing is that you can do many things with one pot of samples. And it's really expanded from there. And we're very, very excited about the potential for this work.
Thank you, Tom. Ilana, please.
I was going to add that, so like Tom described, coming into my graduate work, it was kind of the big picture goal that we were always working towards. Like Emily had mapped in a proof of concept focused on the secretin family of GPCRs, their interactions with the 3 ramps. And my goal was always do it across all the subfamilies, across the whole GPCR superfamily phylogenetic tree. So I think in one regard, I was really lucky that I had that kind of guiding, very specific goal from day one. But then in addition to that, we wound up kind of adding in a lot of layers that turned out to be, I think, equally meaningful, including the tool development, which includes the construct and the antibody validation, and also moving from looking at overexpressing GPCR-RAMP interactions to trying to identify the natively occurring ones, and then adding in also the additional dimension of validation with proximity-based assays. So we were kind of always shooting for the map the entire interactome goal, and it wound up kind of becoming much more rounded out.
I love it. And, you know, Tom, when you mentioned Stephen Ford, I do remember last year when we were at the GPCR retreat, Kathleen Caron's talk as well. And she also mentions actually picking up the phone and calling Stephen and asking him for the constructs. And I also do remember meeting Kathleen for the first time, where she was working with Tracy Handel on getting to understand how do ramps, or if ramps at all influence chemokine receptor activity. And of course they do. What doesn't? But I thought it was a really interesting subject and a really interesting set of proteins. And to your point, Tom, these are single transmembrane domain proteins. GPCRs have a wide variety of physiological effect. They signal in many different ways, and you need that diversity. And I think ramps build that diversity of signaling in there. So tell us a little bit more. Oops, I think we lost Tom there for a minute. But Ilana, tell us a little bit more about what are these tools that you have developed, and how can the GPCR community benefit from these tools? What are the questions that people can ask that they couldn't ask before you developed these tools?
Mm-hmm. Yeah, so I think we can look at the tools from 2 angles. The first is digital tools, and the second is physical tools. So on the digital tool side, and this is a lot of effort from our collaborators in the Jochen Schwenk lab, particularly a graduate student named Leo Dahl and Annika Vendes. We developed a web interface where you can go in with your favorite GPCR and see the results that we found from our multiplex assays. One, in terms of the antibody validation for the different GPCRs, and 2, in terms of the GPCR-RAMP interactome. And so the idea is, if you are studying a receptor, a subfamily of receptors, you can use this online resource to easily identify tools, namely the antibodies that could be used to study them, because as we know, it's very hard to generate specific anti-GPCR antibodies. And also in terms of studying the signaling, the behavior of the receptors, you could see if they were identified as potentially interacting with the ramps, and if yes, which ramp, and that could guide the biochemical characterization of those receptors. So that's on the digital side. On the physical side, we have a library on Addgene of DUET GPCRs, where the DUET stands for dual epitope tagged GPCRs that we designed and used in our multiplexed screen for the GPCR-RAMP interactome. And these receptors are based on the Presto Tango GPCR library of Brian Roth, and we engineered them to have epitope tag on a flag tag on the N terminus and a 1D4 tag on the C terminal tag. And you can order them, use them for a wide range of applications that you can imagine with these epitope tags. Especially 1D4 is particularly selective and gives great signal in a lot of different applications.
I love it. I love it. And you make milligrams of it in Tom's lab.
Yeah, I want to give a shout out as follow up to Addgene, because Addgene's a great resource, and when we reached out to them originally, we said we want to deposit 220 clones. They're all related, but they're going to be separate. And some people may want one, some people may want all 220. And they were really amazing. It took a lot of work on our end, a lot of spreadsheets and 96 well plates transferred back and forth. But I think it's really a great resource for the community. And it's somehow coordinated with these web-based resources where all the data is available freely, and the constructs that relate to those data are available freely. And this is really something that I give Ilana a lot of credit for pushing this over the finish line. But we're very happy, and something like already approaching 500 people have requested clones from this Addgene library, so we're very happy about that.
It's really remarkable. And I did have the opportunity to work with Addgene when I was at IPI, and actually we were getting ready with them at the time to release our research use only antibodies. And it takes a lot of work, but they're at Addgene, they're very well organized, they know what they're doing, and they have their processes up and running and set up to help the community for sure.
Yeah, I think these central resources like Addgene, maybe like when you're at the bench, it may seem less glamorous to like work on this sort of tool development, but I think this is really kind of a catalyst for so much research that actually winds up affecting so many different lines of investigation.
Exactly. And can you tell me if at Addgene these tools are available for academics and industry or only for academia? I don't know if the depositor determines that, or is it Addgene that determines that?
Well, they're certainly freely available for all academics who want to do research. There is a small fee for clones, which we don't benefit from, our lab doesn't benefit from, but it's a service fee from Addgene.
Yeah.
And that fee, of course, increases, but is economical if you wanted to get the whole library. You wouldn't have to pay 217 times whatever the fee is. There would be a smaller fee for the whole library. And commercial entities can access that, but it's a different kind of a licensing program.
Yeah, if you were to just go on Addgene, it would only, like addgene.org is just anyone going on their browser, it would say only available to academics and nonprofits.
Okay.
And the fact is that if a company wanted to use these things, they would probably have the resources to just make them themselves.
Yeah.
And what we're trying to do is help all of us, all of the like-minded academics out there are just trying to push things forward without worrying too much about the commercial applications.
No, I
But
agree.
yeah, it's
I see
really
it. I think it's
yeah.
fantastic. 215 results just by typing in Tom Sakmar. It's really fantastic. I love it. I love it. I can't wait to dig in and get a sense
But
of.
it's not, the thing, Yamina, that you have to realize it's not only ramp receptor interactions, because you could use these constructs, or you could ask a question: does my favorite scaffold protein in the cytoskeletal assembly interact with GPCRs? And you could use these constructs for things like that. We just happen to focus on ramps because of our original interest in family Bs, going back to working with Debbie Hay and Harriet Watkins and the other folks going back 20 years. And it turns out there are a lot of things happening. It's not only heterotrimeric G proteins and beta arrestin. There are other important proteins that interact with GPCRs, and this is a potential way to take a look at some of those questions.
Yes, and including other GPCRs in terms of heterodimerization.
Then you both make a great point. That is true. We keep on being focused on the RAMP, the G protein, the beta arrestin, and maybe heterodimers, as you mentioned. That's pretty much it. But we don't spend enough time, and maybe it's because we don't have the right tools yet to detect interactions of GPCRs, G proteins with other intracellular effectors that actually do modulate our GPCR function.
Yeah, and I'll just mention a couple of them that we have looked at and are interested in. One is this, they're both scaffold proteins. One is called 1433, another one's called P11. They're involved with different activities in different tissues, and there's some disease associations with these interactions with GPCRs. So I think there's a lot of work that can be done. We take the sort of more biochemical approach, but I think these constructs could also be interesting for people who make models, disease models in animals and mice and other things. So yeah, interested
Absolutely.
to help anybody who's interested in this.
Every time I hear the name 1433, I remember my undergraduate class where we first heard about this protein, and I thought it was such a funny name because it's 3 different domains and it's their molecular weight. And I always wonder, where do people come up with names for these proteins?
Well, at least we're not working in Drosophila genetics, where we make up names to describe the appearance of the mutant flies. But anyway, we're describing the appearance of the mutant molecule.
Exactly.
Anyway.
I'm going to be dragging because I do have my Rockefeller University cup, not my regular Dr. GPCR cup with me today, in honor of us being all connected through Rockefeller. I wanted to ask, so now you have the online or the dry lab tools, you have the constructs. And Tom, you also mentioned that people could be using these constructs that are available at Addgene to study the interaction of any of those 215 GPCRs with other accessory proteins, other proteins. What are some of the questions you think these tools, or how are these tools going to help the community, in addition to obviously looking at RAMP interactions with your GPCR, any other protein, any other ways the community can benefit from these tools, either directly or indirectly?
Maybe I could start out, and then maybe Ilana could fill in whatever I miss or make a mistake about. But I'm very excited about these observations that autoimmune diseases and infectious diseases have a component in which there are some autoantibodies generated in autoimmune diseases that actually recognize GPCRs. So these would be GPCR autoantibodies that are part of the molecular pathophysiology of autoimmune diseases. And remember, endothelial cells express a large number of GPCRs, and they're exposed to these immune reactions. And it turns out that there are classic autoimmune diseases in which it's known that anti-GPCR antibodies play a role. But more recently, including in things like long COVID, there have been suggestions that auto-GPCR antibodies in disease states are part of the pathophysiology of those diseases. And really interestingly, it turns out that many of those antibodies, you'd think that if there's an antibody against a GPCR, that it's going to block the activity of that antibody. But not so fast, because some of those autoantibodies actually are activating autoantibodies. The antibody binding to the receptor causes signaling through that receptor and pathological signaling. So we think that some of the tools we've developed would be useful for the discovery of autoantibodies in human disease states. Basic research, but also clinical diagnostics, because existing tools, you say, well, okay, maybe there's a antibody against thyrotropin stimulating hormone that causes Graves' disease and thyroiditis, and you can buy a kit for that antibody. But what about the other 200 potential autoantibodies that are raised against GPCRs in these non-specific illnesses? I think there's a lot of room for basic research that leads to clinical diagnosis. And even some of the antibodies that are activating antibodies against GPCRs could even be disease treatment leads. An antibody that activates a receptor could be a lead antibody for a therapeutic implication. So, yeah, I'm very excited about the future of these biologics targeting GPCRs, not only small molecules.
I love it. And to your point, it's very difficult to find or to discover antibodies that activate GPCRs. Finding a blocker, I wouldn't say it's a walk in the park, but almost. But finding an activating GPCR, an antibody that activates a GPCR, and I'll push it, maybe finding a biased antibody would be even more cool.
Yeah, I think to build on what Tom was saying and what you were saying, Yamina, that this assay, it could be, the level of complexity can be increased from looking at, okay, can we identify GPCRs, for example, that have autoantibodies that bind to them in whichever disease state, to then try to figure out, okay, what are the functional consequences of these antibodies that we've identified? So kind of like GPCR to antibody, but then you could draw the arrow back and see what the effect is. You can think about adding ligand into that system, seeing how that affects or disrupts or modulates that interaction. So there's really so many variables and components to the system that can be added to gain more and more granular understanding. And I think I really want to emphasize that it can be both for basic research, like understanding a particular disease state or a healthy state or cell type or tissue type, but also with really, I think, clear and exciting like diagnostic implications, because right now, most of these autoantibody tests are in ELISA format, which is kind of tried and true, but not exactly multiplexed.
Yeah, and I think having an assay where you can multiplex, where you can collect a lot of data, allows you to do it, as Tom was mentioning, in a small tube and collect lots of data. And obviously having a process that allows you to deconvolute all the data you're acquiring allows you to very quickly get to those answers and then plan on the next experiments and what are the key experiments and how it's really spinning it up. It's like doing, a Western blot, and I think maybe this is going to reveal how long I've been in the field, measuring cyclic AMP production and purifying your radioligand cyclic AMP on 2 sets of columns, and then taking your tubes to counting, or running the assay in a 384 well plate and running many compounds or many conditions in one plate in an afternoon, compared to days of work.
Yeah,
Well,
I think
it's really great, Yamina, to try to date yourself when you're on a call with me, because no matter how far back you go, I'll always be able to one up you. So don't worry too much about that.
Yeah, well, I was talking about the columns, and I do specifically remember having the racks ordered at some kind of a shop where Michel Bouvier also worked with whoever did those racks, and then the columns were filled with the resin, and someone in Michel's lab had a tube attached with to a little stick, and the tube was calibrated in a way that in that little tube you could measure exactly the number of grams of whatever resin you needed to put in there. I honestly, I think I still have it somewhere. My husband should not be hearing this, but I keep all sorts of weird things, and this is one of those little tools that should be in a museum by now.
That's really funny. Well, of course, CysBio has really helped the field by developing these fluorescence-based approaches to do something similar, and we don't have to use radioactive tracers so much. I mean, they're still useful and shouldn't be ever abandoned, but it's a different world than it was.
Absolutely. So, Ilana, you were mentioning that. So, before we hit record, you were talking about this particular project, these particular results. But what next, from a tool development perspective? What's going on? I know you're no longer in the lab, but as you were leaving, what were the ideas?
Wait, you're no longer in the lab, Ilana? What happened?
Maybe we could talk about it more. But yeah,
we know Ilana's moved on to better things, but is still a part of the lab family. And I'm really curious to hear what her perspective is on this project.
Yeah, I feel like, so maybe I could start, but Tom could give the real insight into what's currently going on. But when I was leaving the lab, I had the pleasure of kind of working with and mentoring a little bit, of visiting a student from Germany and a rotation student who has since joined the lab and is very interested as well in GPCRs and ramps, which I hope I can take even just like a little bit of credit for. That would be really nice. Who was interested in continuing this project, and she has a really unique, more computational background. So I think one of the directions in terms of GPCR-RAMP and the tool development, in addition to what we were just discussing in terms of autoantibodies, is also the computational aspect. Of course, like one kind of wish that has always been in the field is to use computational tools to predict or describe GPCR-RAMP complexes, which has not been possible before. But now, with a lot of new developments, including with things like AlphaFold, what we can computationally do, that horizon has expanded dramatically. So I think that is one direction that the GPCR-RAMP project, so to speak, is heading in the Sakmar lab.
Yeah, and I'd like to say that we really are happy that Ilana has moved on to such a great position still in science, but not so much bench science anymore, but still making a difference. And she was able to train some great students, including Yangwan Chen, who was part of the recent papers that came out, and he's continuing on with this work. And Darius Rausch from Germany, from Peter Hegemann's lab, did a 6-month rotation on this project. Margaret Wong is a new graduate student, and Ariel Pan is another graduate student. And they're all really fascinated by this project and this concept of multiplexing to look at protein-protein interactions. Very few students nowadays, maybe this is an editorial comment, but very few students want to study, at least in molecular biology, biochemistry, chemical biology. They don't want to study one particular protein-protein interaction, for example. They want to use technology to develop, biosensors or chemical readouts or multiplexing technologies or miniaturization. And all of these things are so important for the field. And I think we're really fortunate that Ilana set the standard for that.
I love it. Ilana, you were going to say something else? No, okay. I thought I heard you. No worries. No worries. Not at all. No, I think
she's speechless because of all of this fantastic work, and it really has set the stage for a lot of I think important future work in the field. And by the way, our collaborators in Sweden, we shouldn't go through without mentioning them, because Jochen Schwenk at SciLife Lab, Matthias Sulin at the Royal Institute of Technology in Sweden, the Karolinska Institute, Per Svenningsson, the student of Per Svenningsson, Marcus Saarinen, who visited the lab. We could not have done this project without this amazing core resource in Sweden that we happened to connect with, because Emily Lorenzen, going back to the beginning of this podcast, reached out to them and said, hey, could you help us? And they said, we don't know really anything about membrane proteins, but maybe we should. And we said, yeah, work on membrane proteins. The serum proteins are easy. And Atlas Antibodies, they said, we have 18,000 anti-protein antibodies, and we have about 1,000 of them are membrane protein, but we haven't characterized them. And we said, yeah, you should. Let's work on methods to do that. And I think it's always good to think about what's possible and to get collaborators. It's an international, despite what we hear nowadays, the future is still globalization of science.
Yeah.
And there's no science that ends at a national border. Let's just get that out there.
And I think that's an important point to make about science not being, limited by borders nowadays, and not being limited by the number of wells that you can run. And doing an ELISA, you can do multiplex, you can acquire lots of data, and you could bring in the computational aspects of it in order to potentially find relationships between your proteins that the naked eye, the poor human brain couldn't see unless you would spend years and years on it. And now with I'm exaggerating obviously, but with the click of a couple of buttons from someone who actually knows what they're doing, you can map your molecules, your interactions, and you can connect those together and see islands that may be of interest.
And tying it back to what Tom was saying about how he started with studying one, now there's kind of a trend towards studying many, and that's obviously what we were trying to do in terms of the GPCR-RAMP interactions. And I think one other direction is kind of being able to use this information on many to go back to one in a very focused and refined way that would not be possible otherwise. And just to give two examples, one thing that I always, if I could do 2 PhDs in parallel, the second thing I would do would be to use the GPCR-RAMP interactome results to try to deorphanize the significant number of orphan receptors there are, which would be another kind of screen type of project, but then that would become very focused on the receptor level. And then the second thing is the lab started with this interest in the class B, And then that led to GPCR-RAMP. And this kind of leads back to class B, because now with the GLP-1s, this is kind of which GLP-1R, we and others have shown interacts with the RAMPs. It kind of is a really great example of how understanding this at the superfamily level can then be used to really zone in on specific GPCRs that are very relevant for different disease states.
No, absolutely agreed on this. I actually really like the idea of trying to deorphanize using this system, because it's hard to work on a protein or a GPCR where you don't know where it's expressed. Can you detect it? Do you have an antibody for it? Do you know what it does? And do you have a compound or a ligand that you know works? And so one nice way is to really look at its interactions with other proteins, including the RAMPs.
Yes,
wonderful.
Exactly.
I love it. All right, we agreed on about a 40-minute podcast. Tom mentioned in the beginning before we hit record, you're going to hate me for this, that a 40-minute podcast is the right length for the podcast. Any last words before we wrap up?
I would say the last word from me would be thanks to you, Yamina, for your perseverance and setting up this fantastic network, and including us in these discussions, and keep up the good work.
Thank you so much. I really appreciate it. And thank you for spending the time to talk to me about all of these new tools. I will obviously reach out to make sure that I have the right website, the right references, and everything is going to go up on our website. I'll have the team to send you a link to our Google form. We already have your pictures, bios, but anything additional you'd like to change, we're more than happy to do it. And then people will know where to find you as well.
I'd also like to echo what Tom said, and thank you, Yamina. This is like an amazing platform, Dr. GPCR ecosystem overall, and of course also the podcast. And I also want to say, Tom gave me a lot of hugs during this chat, but really none of this would be possible without him and his lab and the type of open and supportive culture that he creates.
Thank you so much, Ilana.
Thanks, Ilana.
All right, don't go anywhere. I'm going to stop recording. We're going to say goodbye a little bit more informally and let people wonder what we talk about after we stop. I always do this. If you listen to the podcast regularly, I always do this at the end, but I still find it funny. So I'll keep doing that until I come up with something new. All right, see you in a bit.
So long.
I need a favor. If you enjoyed this episode, please click the like button. And if you like the podcast, consider subscribing to our YouTube channel. Many of you come back to watch our videos but aren't subscribers yet. One click goes a long way. All the information about Dr. GPCR is in the description. Find all our fantastic features and everything you can benefit from becoming a premium member. And while you're on it, check the Dr. GPCR University courses and share them with your network, colleagues, and friends. We also want to hear from you. Is there anything missing from our ecosystem? Can we make it better? Email Yamina at drgpcr.org or email our team at hello@drgpcr.org. Thank you so much for joining us in this podcast episode. If you're still there, we also want to take a minute to thank our team members: Yamina, Attila, Montserrat, Cam, John, Yatsu, Mark, Ivana, and Andreina. If you like our podcast, you can leave us a 5-star review on Apple Podcast. Until next time, and stay safe.